then we are on the final experiment of Module 3, which is the DNA extraction lab. Now, before you begin this experiment, you're going to need to buy some fruit, and I highly, highly recommend the strawberries. You can use them frozen or fresh. I would not get the grapes and I would not get the bananas because the banana is really hard to see and the grapes are just a little too light. So strawberries work the absolute best. You don't need that many either. Like 1 or 2 will work just fine. You're gonna also need a small plastic Ziploc bag, which you'll have to provide. Now the things that the kit provides is this 10 milliliter graduated cylinder. You will need the DNA extraction solution. You will need the ethanol solution, the 70% ethanol solution. A standing test tube, which is what came with the cap in your lab kit. A wooden stirring stick. And the cheesecloth that came. Now, before you begin this experiment, you have to chill the ethanol. It has to be in the freezer for 60 minutes. So put that in your freezer, wait 60 minutes, and then you can start. It has to be cold or else it will not work. For about an hour. Now don't worry if you forget about it and leave it in there longer than an hour, it's okay. It's not gonna freeze. You just want it really, really cold because this helps with extracting the DNA. So we have that, and then we have our DNA extraction solution. And you're gonna need some strawberries like I mentioned before. Don't do the grapes, don't do the bananas, it doesn't work very well. You're gonna also need your small graduated cylinder, a standing test tube, so those test tubes that came with the cap on it, some cheesecloth, and a wooden stirring stick. So the first thing you want to do is, after your ethanol has been in the freezer for an hour, you're going to go ahead and take just a couple of pieces of strawberry. You don't need very much at all. Like 2 or 3. Frozen works just as well as fresh. Put it into a Ziploc bag. And then you're just going to start mashing it with your fist. Just squishing it. I used to do this with some other students and they really found this therapeutic. You can also do it with your kids. Mine went to bed already, so they don't get to participate right now. Alright, so once you have kind of just squished that to a pulp, you're going to go ahead and move on to step 3, which is pouring your DNA extraction solution into your graduated cylinder. You just need 10 milliliters, which is the top part of the graduated cylinder. So just keep your eye on it. And there's not too much of this solution, so it may actually take the whole bottle. Maybe not. Yep. All right, well, that is the whole bottle. So don't mess this up. You won't have anything to redo it with. Okay, so then you're gonna pour that entire solution into the bag. Okay, seal it, try and get all the air out. Okay, and you're just gonna knead it for about 2 minutes. This just kind of works works it in. This DNA extraction is really important for separating the DNA from all the other parts of the strawberry. Okay. So, next thing you're going to do is you're going to take your cheesecloth and you're going to just, this cheesecloth that I have is the 15 centimeter one. You don't need that much of it. So I'm just cutting a little bit. And then what I like to do is I unfold it and then I just fold it over once so that you just kind of have a double layer. And you set it over your test tube and then you just secure it with a rubber band. And I don't have a rubber band, so I'm using a hair tie, which they have elastic in them, so it works just as well. You want to secure this pretty well because you're going to be filtering your solution with that. And so now we're going to take your bag and kind of move the strawberry solution down because you're going to cut a hole in the bag. So we're just going to cut a little hole. And we are going to pour the solution through the cheesecloth, and it's going to be messy, so if you spill, it's okay. It's not the end of the world. So I mean, you could put a plate under this, or you could put paper towels, whatever you felt like. Don't try to pour it all at once, because see, I kind of made a mess there onto my ruler, and that's okay. And so then we're just gonna let it take a minute, and then we're gonna pour some more. And if you only end up with a little tiny bit, that's okay. You don't need a whole lot of it. Now I'm just gonna go get something really quick to sop that up. There we go. Okay, so not much is coming out, but that's okay. I'm gonna get I'm gonna change out this cheesecloth. So I'm gonna get to where it's not doubled over, and instead where it's just one. Because I think it's a little too much. So let's try it with just one. Okay. And the DNA extraction material is not gonna hurt you, so you don't need gloves. Okay. Alright, so going back through the hole again. There we go. Okay. So that's working out a lot better. So go ahead and just make sure that you're only using one layer of cheesecloth and not two like I started with. And so you'll get these chunks that sit on top, and you can just kind of wipe them off because we're more interested in the liquid portion. All right. So I'm just going to move these chunks off the top. Okay, so once you've poured you've filtered all your liquid, you're just gonna let it sit there for a minute because the next part you have to rinse out your graduated cylinder. So I'll come back whenever I've done that. Okay, so I've kind of cleaned up my area a little bit, and I rinsed out my graduated cylinder. And when you rinse it out, it's going to be like really foamy. So just make sure you get all of that extraction out of there. And whenever it stops like foaming, then you've gotten it out. So you can see that you only get like a little bit. You don't get, you know, like a full test tube or anything like that. You would need to do more strawberries for that, but just this little bit is okay. You'll still be able to see what you need to see. So I'm gonna set it there. So now we are on step 7. So now you need your ethanol from your freezer, and you're gonna measure out 5 milliliters of it. So you're just gonna slowly kind of pour it down the side until it reaches the 5. Okay. Right, so then what you're gonna do is you're gonna hold your test tube that has your strawberry extract in it, and we're gonna slowly, so we're gonna bend the graduated cylinder kind of like at a 45-degree angle and slowly start to pour to where the ethanol is sliding down the side, and you're not just like dumping it all. So you just want it to gradually flow down the side until it's getting into the tube. So what you should end up with is a layer of ethanol on top. And what starts to happen, I'm probably holding it too high, sorry. So this is what it looks like. What starts to happen is you start to get all this stringy material coming up. And that is actually the DNA of the strawberry. It kind of looks like this slimy residue. And so what's happening is that you want this to kind of sit for, 2 to 5 minutes, and the DNA will precipitate out of the solution. So that means it'll come out of the strawberry extraction. And you're going to start to kind of see like air bubbles and things like that. And what should happen is that the DNA will kind of start to float up to the top, and you'll see strands of it. So we're just going to let this sit here for a minute while I go to the next page of the instructions. Okay, and the last part is kind of fun. You take the wooden stirring stick, and you are gonna kind of dip out the DNA. Now, if you don't get DNA, it's okay. It's not a big deal. You can kind of look at what happens here for your lab, but you should get it. That's why I said to use strawberries, because it's really easy to see the boundary. So I'm just gonna kind of dip it in there and show you. But yeah, all this kind of like white stringy stuff, and I don't know if my camera is picking it up well or not. That's DNA. So that's pretty cool. Most people like this one. All right, that's it.